Okay. Yeah, so this course is called Colloids and Surfaces. Um, so you will see that there are, okay, and so before I begin, why this course is kind of called Colloids and Surfaces. Uh, so what I'll do is I'll first give you a okay, so this is the course book that I'm going to follow. Okay, so that's a picture of the front cover, and this textbook is titled Colloids and Surface Chemistry, which is written by Hemans and Rajagopalan. Okay, it's a good book, very basic. Okay, I have learned a lot of things by reading this book. That's going to be the course textbook. I have a copy of it in case if you want to take a look. Of course, library will also have a copy in case if you would like to go and refer. Apart from the course book, I have a lot of reference books listed here, about 4 of them. Some of them are advanced, some of them are very basic. Okay, you can take a look at them, in case if you would like to refer to some specific topic. Okay. So what I'll do is I'll begin with talking to you about introduction to the course. Okay. The aim of today's class is to essentially define what is colloids. I'll just do that. Followed by I'm going to give you several examples, where we have we use colloids in different fields. Okay, so that's kind of that is to motivate you from learning colloids. Okay, so I'm going to take out some examples from different fields and then tell you how the colloids principles are exploited to make some new materials and things like that. Okay, that's okay, so that's a long list of what I'm going to cover in the maybe next few lectures, but for today most likely I'll just define what is colloids, and then I'll follow it up with talking to you a little bit about motivation to study colloids. Okay, why are we trying to look at this colloids, and if there is some time I may look at something on classification of colloids. Okay, that's for today. Now definition is a textbook definition you will see in a lot of forums as well. A colloid, okay, is any particle, okay, which has at least one dimension in the size range between 10 power minus 9 meter and 10 power minus 6 meter, that corresponds to 1 nanometer to about a micrometer particle. Okay, so the moment you have any particle in this size range, that's what is called as a colloid. The reason why you have at least one linear dimension is because often you may be working with particles that are spherical. Okay, in such a case you need only one dimension to characterize such particles. The moment you think about other shaped particles, it could be ellipsoids, it could be cylinder like or rod like. Okay, then you have more than one dimension. Okay, so if I take a 2-dimensional ellipsoid, you need the length of the major axis and length of the minor axis, okay, to define the size of the particle. So therefore in such a case, if one of them, in this case B is smaller, as long as B is in the size range from 1 nanometer to 1 micrometer, that is still called a colloidal dispersion. So as long as one of the linear dimension of the particle that you are dealing with is in this size range, that is what is called as a colloidal dispersion. Okay. Now it turns out that a lot of materials that people work with, okay, and a lot of materials that are biologically important, for example DNA, virus, proteins, okay, and a lot of consumer products which have for example detergents and shampoos which have surfactants, right? So all these molecules, okay, when you put them in solution, when you put them in water, they form aggregates, okay, whose size again comes in the size range between 1 nanometer to 1 micrometer. Okay, or you could have particles present in some fluids, for example milk. Okay, milk contains some particulate matter. Again, the size of these particulate matter is in this 1 nanometer to 1 micrometer size range. Therefore, there is a lot of relevance looking at colloids in many different fields. So, this is a triangular diagram. On one apex you have polymers, the other apex you have particles, solid particles, and the other apex you have surfactant molecules, which are also called as amphiphiles. Okay, and several different things are between, right? This DNA, polymers, blocker polymers, proteins, right? So, all these materials, okay, when people are working with them in solution, they invariably form, okay, aggregates of 1 nanometer to 1 micrometer dimension. Therefore, there is a lot of interest in looking at such systems. Okay. So, just to give you some examples again, I have taken a few examples from again biology. So, what you are looking at is, this is an image of a virus, is what is called as a FD virus. Okay. And so, this FD virus has a length of about 800 nanometer, and the diameter, this dimension, right, it is about 6.8 nanometer. Okay. You can think about that's an example of a rod like a particle, right. So, people are working with such virus particles. If you put them in fluid, in water for example, they form a nice model system to look at rod like particles in water, okay, or a dispersion of rod like particles, right. And another example again from biology is this E. coli, which looks elliptical in shape, right, and that's a scale bar that you have here, and that scale bar is about 5 micrometer. Okay. Therefore, if I look at the dimensions of these things, again they are in that 1 nanometer 1 micrometer size range. Okay. So therefore, these are again classic example of colloids, but in the field of biology. Okay. Now, I have another example here. So, what you are looking at here is a polymer. It's an example of what is called as a blocker polymer. Okay. So, go back here. So, this is a schematic that you see. This is of a polymer. So, typically people show polymer as some kind of a chain, right, which basically has several repeating units which are connected by some covalent bond. But you could have a system where I can have two polymers which are connected by a covalent bond, as an example that is given here. So, that is orangish color is one type of polymer, and the blue is another type of polymer. Again, they are connected by a covalent bond. Okay, that is an example of a blocker polymer. You have two different polymer molecules which are joined together, right. That is why it is called as a di-blocker polymer. You can have 3 different chains, something called a tri-blocker polymer, and so on and so forth. Okay. So when such polymers, which are again represented by the pink and the blue color here. So, when you take such things and put them in water, they again form different structures. They could, again I am going to introduce these terms a little later, something called as vesicles, some kind of a spherical aggregates. If you again look at these images here, if you look at the scale bar here, again 200 nanometer here and 50 micrometer. So, the moment you put these things in water, they automatically assemble, okay, what is called as a self-assembly. They self-assemble to form these aggregates, and these aggregates have a dimension 1 nanometer to 1 micrometer. So, there is a lot of interest in looking at such systems as well. Okay. Now, to again further motivate you why people are looking at colloids, you will see several examples where people talk about drug delivery. People are using some kind of containers for drug delivery. One common object or one common kind of a container which is used a lot in such application is something called a liposome. I do not know how many of you have heard this term. Lipo means lipid. Okay. So, this is a spherical structure. It looks like a sphere. Okay. Again, example was shown here, right. This is an example of a structure very similar to liposome, but it is made by polymer. Therefore, it is called as a polymerosome. Okay. Similarly, if you make such structures which lipids is what is called as a liposome. So, the way these structures are made is you take a lipid molecule. So, lipid molecules are represented by something like this. They have a chemical group which is hydrophilic, okay, and it has some group which is hydrophobic, okay. Typically lipids have one head group which is hydrophilic, okay, and two tail groups which are hydrophobic. So, when you take several such molecules, okay, in water, and what I do is I simply mix it up, okay. I just put in a sonicator or or a high energy mixer. I just mix it up. In the end, what you do is you basically get structures which look like spheres, okay, and the inside, this is, you can think about this as a core shell kind of a structure, where the core has water, because the surfactant molecules or these lipid molecules were put in water, okay, and inside has water. This region here, okay, the shaded region is actually this layer, okay, and that layer is made up of what is called as a lipid bilayer, okay. What lipids do is they kind of arrange, in a kind of arrangement like this. There is one layer of lipid, there is a second layer, that is what is called as a lipid bilayer, okay, and these lipid bilayers fold to make these spherical structures, okay. So, what you are looking at here is a cross section of one such liposome. If you cut it up, okay, what you see is these lipid molecules are nicely arranged in a bilayer fashion, okay. Now, because the tails, okay, because these are hydrophobic, right, these lines that you see here, they are hydrophobic, I can use this region for incorporating a hydrophobic molecule. For example, what is put here is what you are looking at, this elliptical looking like thing is a hydrophobic thing that can happily sit in the bilayer region, okay, and I can have this hydrophobic part which could be happily being in water. Therefore, I can use such containers to either carry hydrophilic material or a hydrophobic material or any complex structures which can be there both in the hydrophilic part and the hydrophobic part. So, you can actually use them as cargo carriers. That means, if I take such things and if I make such formulations and give it as a medicine, for example, okay, I can put both a hydrophilic drug and a hydrophobic drug in such things, and I can easily transport them into the body, okay. So, therefore, people are looking at such colloidal structures as cargo carriers for applications in cosmetics, medicine, genetics and things like that, okay. So, this is one one of the motivations why one would be interested in looking at colloids. This is an example where people have taken surfactant molecules or lipid molecules and made structures which are colloidal in length scale, and then looking at some of these interesting aspects, okay. Now, to come to a second example of why should one look at, I have taken some examples from materials where there is a specific field called colloidal processing of ceramic materials, okay. So, ceramic materials, one could be interested in looking at SiO2 or aluminum oxide, or any such system, and one of the ways of making such materials is by using what is called as a colloidal, colloidal processing route, in which what you do is you start with a uniform, you start with the particles, okay. I have particles, I could buy them as like a powder form, okay, and it has uniform particles of exactly same size, okay. Now, what I do is I take such particles, I put them in a fluid, okay, to make what is called as a sol, okay, which essentially is a dispersion of particles in a fluid, okay. That is about it, okay. Now, what do I do from the sol? Now, what I do is I have a sol, and from this sol, if I know everything about the particles that are present in the sol, I am going to talk a little bit about a few things later on in the course. If I know what is called as how the particles are interacting in the fluid, Again, as I said, I'm going to talk about these things a little later in the course, okay? But at this point, what you should remember is that I have something like this which can easily flow, okay? It has fluid plus particles. I have this, that's my sol, and I take exactly the same thing. I do some simple manipulation to go from such a state to something where things don't flow. Okay, this is an example where I have gone from a sol where I have a nice dispersion of particles into a kind of a solid like thing which doesn't flow anymore, right? And I'll tell you how we do that in the meantime. And now that's an example of what is called as a wet gel, because it still has a fluid, okay, and the particles that I have in the sol are kind of, they form a nice network, okay, and this network spans the entire volume, okay. I can go from one end of the sample into the other end just along the particle network, okay. That's what is called as a wet gel. Now what I do with this, I take this and then I evaporate the fluid to get what is called as a dry gel, okay. And you can do other processing. I can actually do what is called a compaction. If I want a porous material, I can process this. If I want a more denser material, I can go to this route, okay. In the end, what you do is I take this and I do what is called as a sintering, okay. Sintering is a process where I take some sample like this which has particles, and then I heat that to a very high temperature close to the melting point of the particles, and what this does is it facilitates what is called as a solid state diffusion, because of which the particles can form a nice bond, okay. And once that bond is formed, I finally have a hard ceramic material, okay. So that's the final specimen that people have made. Either I can get a final hard specimen like this, or I can also get a porous material like this, which will essentially have all the voids that you had in the sample. I can do that just by sintering process, okay. I can either do this, or the other way of doing this would be I take the sol and I deposit a small layer of fluid on a substrate, okay, and I let the particles deposit on the substrate, and I can still sinter it, and I can also get a thin film, okay. So there's a lot of interest in looking at such studies where, in order to do colloidal processing, where you start with a powder, make sol, gel, and ultimately ceramic material. So all of this can be done if you have a really good understanding of colloids principles, okay. So what are colloids, how do I go from a solid state to gel state, okay, how to handle these materials, right, things like that. So another example, again I'm going to talk about some new material development. So what you're looking at is, they look like spherical objects, right? They look like spherical objects to you, and that's 250 micrometer, right, really large, right. So therefore these are actually droplets, okay. These are drops. How these were made? By a simple experiment. What you do is I take water, okay, and I have an oil. I put in particles. So in this case, this contains about 2.2 micrometer, positively charged particles, and a little larger, this is 9 micrometer, negatively charged particles. You take that and you have oil, and you just simply mix it, that's it, okay, that's it, right. Now what you do is you create what are called as emulsions, okay. When you do that, you make emulsions. In this case, the emulsion that formed, they are water droplets in oil, Now whatever particle that you had in the fluid, they go and sit on at the interface, okay. So what this is a droplet like this, and because these particles are big enough, right, 2 micrometer and 9 micrometer, they are big enough, I can do microscopy. If I look on the droplet surface, what you have is this nice arrangement of 9 micrometer particles and 2.2 micrometer particles, okay. You are able to emulsify two fluids which otherwise don't mix, I'm able to make an emulsion by using particles. Okay. So this is an example of what are called as particle stabilized emulsions, also called as Pickering emulsions. Okay. Pickering, because Pickering was a scientist who found this for the first time in the 1900s. Okay. Now you can ask me what is the use of such things. So I'm going to show some examples. So this is I talked about liposome, right? I talked about polymerisomes. Similarly, these structures that you see, they are what are called as colloidosomes, because this is again a shell which is made up of particles. What you are looking at is a hollow shell. Inside is hollow, okay, and outside has a layer of single layer of particles. Can you see those particles here, right? You can see these nice spherical particles which are very nicely arranged, right? These are actually made starting with particle stabilized emulsions, okay, in which the emulsions are stabilized by single type of particles. I take these structures and then I evaporate the fluid. I can make a hollow structure, and I can this empty space that is within these containers, right? I can again use them for again some drug delivery applications, right? Or I can have several such things. I can compact them and I can do the sintering I was talking about, right? I can take particle stabilized emulsions and I can do sintering, and I can actually get porous materials. Okay, so these are examples of porous materials in which people have taken these Pickering emulsions or the particle stabilized emulsions, and you compact them and you remove the fluid and then you sinter them. People have done metallic materials, porous metallic materials, porous ceramic materials and stuff like that. Okay, and we have another example here which is kind of made by a similar technique. Okay, this is a work of one of the PhD student. So what you're looking at is you can imagine some container like this, okay, which has a Pickering emulsion or particle stabilized emulsions. And what was done is, now what I can do is, now I have these droplets. When the droplets are touching, what I can do is I can do a cross link, right? I can actually connect the droplets by doing some simple reactions. Okay. Now what will happen is, in such a case, I can actually get it looks like a chalk, the chalk piece, right? Piece of chalk, but it's not. Okay, it's a material which is made from some particles, some polymer. Is very soft. I can squish it. Oops, it broke. Not strong enough. Okay, it's brittle, but I can make materials like this, right? This is again done everything from starting with colloids, understanding them and things like that. Okay, so therefore people can make either oil in water emulsions or water in emulsions stabilized by particles, and they can use them for making either hollow capsules for again applications in medicine, or you can make porous materials. Okay. Okay, I'm going to talk about some inspiration from nature. Okay, so what we're going to do is, I'm going to take some examples from nature and then tell you that look, the colloidal scale structures do exist in nature, and there's an interest in looking at such things from that point as well. Okay, any examples that you may have, or do you have anything that you think is in nature and that's colloidal in length scale? So milk was one example, of course, yeah. So anything else? How about this? Do you think there's some connection between this and colloids? Yes, no? Okay, so there's a whole lot of people looking at what is called as structural colors. Okay, so in chemical engineering, right, when people look at things like paints, right, paints of different colors, one of the understanding is that there are a lot of pigments that have a specific color, and you can make paint of a specific color by, right, that's one thing. But there are a lot of things in nature where the colors, the fascinating colors that you see, that comes about because these things have some structures whose length scale is such that, so when the light falls on them, right, light, the light that is diffracted, okay, because of the nature of the colloidal scale structure that are there on these species, okay, that those colors, okay, come about because of, okay, the interaction of light with the way the colloidal scale materials are arranged on such fascinating creatures, okay. Now, so if you look at peacock, right, so these are some, the microstructures that are from peacock feathers, okay. Again, I would like you to look at the length scale, okay, 5 nanometers, 2 micrometer, and look at these rod like things, right. These are about 1 micrometer, so therefore this is 1 micrometer, so therefore the length is about the order of 1 micrometer, and the diameter would be maybe about, less than 100 nanometer, right. So therefore, so this is not the only example. So these are again some pictures of a student of mine who took some years ago. Again, butterfly wings, okay, this is about 20 micrometer, but if you look at these structures here, they're again very much colloidal in length scale, 1 nanometer to 1 micrometer. Plus you have again some example from literature. So these are 2 birds, okay, which have, both of them are bluish in color, right. One is, the light blue, and other one is slightly different, right. Now, if you look at the feathers of these creatures, right, these birds, they have different structures, okay. So this is again the length scale is about 500 nanometers. Therefore, you look at these small dots, okay, that's definitely sub 100 nanometer in length scale, right. So therefore, so the interaction of, the light with such structures whose, size is smaller than the wavelength of visible light, right. So that those interactions is what leads to such colors, which is an example of what is called as structural color, which are not created by pigments, but the way these nanostructures interact with light, okay. Now, so what people do is they kind of use these as inspiration, and you can make such colors in the lab, okay. There are some examples. So this is an example where what you're looking at is 5 different samples, okay. What they've done is what is called a drop casting, okay. Drop casting is a simple technique where you basically have a substrate, and I just put a liquid drop, okay, that's called drop casting, and this has some particles in there, okay. And once the liquid is gone, what you have is a deposition of particles on the substrate, right. The liquid is gone and the particles are deposited on the substrate. So you have 5 different samples, okay, and in all these samples they contain particles which are 226 and 265 nanometer in size. These are polystyrene particles, and what is being changed in the sample from 1 to 5 is the fraction of some carbon black, okay, that is added to the sample, okay. The concentration is given in weight percentage, that goes from 1 weight percentage to 5 weight percentage, and you can clearly see that, the colors are very different, right. And if you look, if you can do microscopy, the microscopy of the sample 3 looks something like this, okay. Therefore, so when you have such drop casting and the particles are finally deposited, okay, so they kind of, you can clearly see from these, it is not a regular arrangement, right. It's more like a amorphous, irregular kind of arrangement, okay. So therefore, so that you're trying to kind of mimic, the kind of structures that you kind of obtain when, there's a amorphous, or not so regular arrangement of colloidal scale structure that are present in such things, okay. You can think about designing such things in the lab, where I take particles of different sizes, slightly different sizes, and I add in an additive, and then I make a kind of a deposit of particles that kind of exhibits different colors depending upon the concentration of some of the additives, okay. So therefore, there's a lot of interest in kind of looking at nature and then mimicking that, by looking at colloid science as a field, and then so one of the motivation could be along these lines. Now, so this is another example of again a common example that everybody gives, right. So there's something called as a lotus leaf effect, okay. That means it's a lotus leaf is an example of a self cleaning surface, right. The moment if you have a a lotus leaf, the moment water drops fall on it, they just roll down, right? And in that process, they take whatever dirt that is there on the lotus leaf, they just carry it, okay? And it turns out that this lotus leaf effect, okay, comes because of what is called as a hydrophobic, not only hydrophobic, what is called as a super hydrophobic nature of the material. That means if I have a lotus leaf, okay, and if I put a water drop, it will, so there are, if I put a water drop any surface, it can take a shape like that, okay, or it can completely spread like a thin film, or I can have a case where the droplet may form something like this, right? Okay, everything depends on the nature of the substrate that you're dealing with, okay. So if it makes a perfect sphere, there's an example of a super hydrophobic surface or completely hydrophobic surface, okay, where the so if it forms something like this, it's an example of a hydrophilic surface, right? Now, so it turns out that the self cleaning nature arises because if you look at these lotus leaf under a microscope, so there are large structures, okay, there are hills, okay, the size of this could be as much as about 10 micrometer, plus you also have this small nanoscale roughness, right? A combination of this nanoscale and microscale roughness is what leads to such self cleaning or super hydrophobic nature of lotus leaves, okay. So therefore there's a lot of interest in looking at these things, understanding again nature, and then try and come up with a way of creating such structures in the lab. And one of the way of doing that is again by a simple technique, okay, which is this is what is done in this particular example. If you are interested, you can go and read up a little bit more about this. Very simple example, anybody could have done it, right? So what you have is a glass substrate. Take a candle, right, and you burn it, and when you burn a candle, you get a soot, right, and you just deposit that soot on the glass substrate, okay, and the soot essentially consists of some carbon particles, right. So the soot, if you it looks flaky, right, the flakiness of the soot actually comes because it has very open network of particles like this, okay. It's not individual particles. The way it comes out, they're kind of connected, they form some kind of a loose kind of aggregates, and so these loose kind of aggregates are kind of deposited on the glass substrate, and I can take that, I can do some simple treatment in the lab, okay. I can expose that to different molecules and stuff like that. So the bottom line is there's a soot that deposited on the particle on the substrate, and if you look at again the length scale is about 100 nanometer, and the size of individual particles would be again much smaller than that, okay. So you have colloidal scale particles, they are deposited on a substrate, and that gives you a contact angle of 165, which is an example of a super hydrophobic surface, right, very close to 180, okay. Not only water, I can create surfaces which have a contact angle of 165, not only for water, also for hexadecane, which is an oil, right. Therefore I can create both water liking substrates or water hating substrate or a oil hating substrates, right. Therefore I can create what are called as omniphobic, okay. These are materials which neither like water nor like oil, okay. So you can make things like that, okay. Final motivation hopefully, okay, is there's a lot of interest looking at colloids also as what are called as model atoms, okay. If you look at atoms or molecules, right, they're sub-nanometer in size, right, they're very small, you can't see them, okay. And we know that anything that is made up of atoms or molecules it does exist in three forms, right, solid, liquid and gases, right. Now of course there have been a lot of developments and people know what these materials are, but one of the ways of achieving all these states the gas-like state and liquid-like state and solid-like state, and visualize them live, okay, is I can use colloidal particles, okay, and I can actually create all these structures, okay, all these materials. So therefore, there's a lot of interest in using colloidal particles, okay, as it is possible to directly observe, okay. I can make a material, I can take something which can go from a liquid like to a solid like state. I can in situ observe how does that happen, how does something go from a liquid state to a solid state. I can observe it, in situ, okay, with doing microscopy and things like that, okay. And how is it done? As we will learn in the course, it is done by tuning interaction forces, okay, the way the particles interact with each other, and it can also be done by playing with particle concentration. Again, I will give you some examples. So this is a something called as a phase diagram, okay, that people first found from simulations. So the number that you are seeing here, it is these numbers. These are what are called as volume fractions, okay, which is defined as the volume of solids or the particles that you have in the fluid divided by the total volume, okay. So all this is telling you is depending upon the volume fraction of the particles in the fluid, it either exists as a fluid state, okay, or there is a coexistence of a fluid crystal, fluid, where there is a disordered arrangement of particles and a crystal like arrangement, okay, or ultimately a glass like arrangement, okay. So this is a phase diagram that was a kind of that was kind of found from simulations, several decades ago, okay. Now, so this is an example where what has been done is experimental visualization of what was predicted by simulation is done through experiments. If you want, you can just look at more details at this page. Now, what can you say about things here, right? This is, so the dark thing that you see, the spherical looking things, these are particles, okay, about 1 micrometer in size. And what can you say about the arrangement here, right? It's very irregular, right? They're kind of, distributed in space. There's no specific arrangement. The arrangement is very much like what will happen if you take a liquid, right? Now, you increase the concentration of the particles. This is about, if you go beyond 0.494 volume fraction, that is more than 50%, a little bit about 50% more, you clearly see that there are regions where there is a crystalline arrangement. Can you see that what is marked there? Every particle has 6 neighbors, very nice arrangement. But of course, there are regions like here where it is still a fluid like, right. So that's a fluid crystal coexistence. You go up in concentration, very periodic arrangement, every particle has 6 neighbors, example of a crystal, right. And if you go further up in concentration, okay, or at some concentration range, you essentially get again a disordered arrangement, what is called as a glass, okay. The microstructure is very much similar to, I can roughly say like glass, like fluid, only thing is the particle concentration is larger there, that's all, okay. The arrangement here and here, arrangement here and here, very much similar. The only thing that is different is the concentration of the particle in the glasses a little larger than what you see, right. So therefore, all that has been done in this example is that they have taken a fluid and you go on adding particles, okay. If I systematically increase the concentration of particles, you see a transition from a fluid state to a fluid crystal state to a crystalline state to a glass like state, okay. And because these objects are micrometer in size, I can do this experiment under a microscope and I can observe these transitions live, okay, in situ I can do it, okay. So therefore, so these are a few different examples from different sources to tell you that look, there's an interest in studying colloids from a lot of different backgrounds, okay, and and I don't know what is motivating you guys from learning colloids, but that you would have to think a little bit and then take this course, so that you'll kind of adapt some of the principles that you're going to learn in this course in your work.